Okay, so let's dive straight back in. So this lesson comes straight on the back of the previous videos. If you've not watched that one already, I suggest you go back and do so within this playlist. So last session we talked about how DNA replicates, what semi-conservative replication is, and so on and so forth. In this lesson we're going to talk about how we know, right? So plenty of evidence base for this. Okay, so how do we know that this is the way that DNA replicates? Why are you just going to believe me, a biology teacher on the end of a YouTube video? This is the evidence, okay? So what we're actually looking at here, right, is three different hypotheses. You've got to put yourself in the position. You're in the 1950s, DNA's just been discovered, the double helix. Watson and Crick have run into the Eagle pub in Cambridge declaring they've discovered the secret to life and all that kind of stuff, you know, however they got hold of that information, so on and so forth. We've now got this structure, this double helix, which raises a huge amount of questions. How does it work? How does it replicate? How did it get there? How do we make sure there's an identical copy in every single cell? Okay, now you've already learned from the previous video how that works. Okay, this is how we discovered how that works. Okay, it's a fantastic example of a piece of scientific investigation that brings in loads of bits of biology that you already know, which is why it's on the syllabus. So there are three ideas. Now, of course, the most logical suggestion, right, was semi-conservative replication from the off. The idea that this double helix structure would split in half and two new strands be made, thus maintaining the order of bases and being the simplest and most logical solution. But just going, that's probably how it works, is not how science works. Science works by experiment and test and evidence. So we had actually three ideas. Semi-conservative was always seen as product frontrunner. Then conservative replication, where the entire old double helix goes into one of the two daughter cells at mitosis, and an entirely new double helix is made for the other. Okay, possible, but it raises another couple of questions. But if that were the case, that would be interesting. And then quite an outlier here, dispersive replication, where the idea is that chunks of each cut vertically, right, if you like, are in one helix and some in the other. Now, that raises a massive question, which is, well, if the base order is really important in coding for DNA, is coding for proteins and making you, and you're just going to willy-nilly cut it, how does that base order get maintained? So that was always seen as an outlier, but you can't rule these things out until you've actually done a test. So the test, Q, Meselson and Stahl in 1958. Okay, so these two guys came up with this incredible experiment, this beautifully elegant experiment to prove which was the correct idea. Okay, prove that semi-conservative replication is the way that DNA replicates. So what they used was two processes. They used E. coli and density gradient centrifugation. So they used different ways to grow bacteria on different agar jelly. Again, something that you all should have done right throughout school. And density gradient centrifugation, which is just a slight modification of differential centrifugation that you've already done in the AS level, right? It would be a standard practical that you've done if you are an A-level biology student, okay? What density gradient centrifugation does, of course, is separate molecules on the basis of their density. So here they are. That's Meselson-Stahl. This is a density gradient down here. We've got centrifugal force from the spinning centrifuge, which again you should all know about. And the idea is that this density gradient is thicker at the bottom, so the liquid is thicker and more viscous at the bottom, denser at the bottom than it is at the top. And the analogy I always give for this is, if you are a person of weak moral character who puts sugar in their tea, it's not really acceptable. It's not necessary. Tea doesn't need sugar in it, so don't do that. But if you do, and you don't stir it, then you'll notice that a load of that sugar builds up at the bottom, and you get a very thin, unsweetened tea, the way it should be, at the top of the tea. And then as you drink down, you get to this kind of gloopy, sticky, sugary syrup at the bottom. That is a density gradient. So a knowledge of this obviously is fantastic for separating out different things within cells. It's also fantastic for cocktail making. And over here, it's a useful, applicable skill. And over here we've got a recent experiment done with some of my A-level students to demonstrate how you can build the density gradient, and they were challenged to make different densities of sugar solution and pile them on top of each other while staining them in different colours. Okay, and this is one such example here, okay, where you've got very dense sugary red solution here, slightly less dense clear solution, slightly less dense green stained solution, slightly less dense clear solution again, and then the lightest, probably just pure water there, stained blue. Okay, now what you could do in that is you could take a material, drop it in, and it would sink to the point where its density matched, okay, and it would go no further, but not push below its own density into the gradient. And that's what we're seeing here. So the large molecules down at the bottom here have been pushed right to the bottom. These molecules here have been sort of stayed sort of somewhere in the middle because they're a little bit smaller, and the smallest one have stayed at the top, making 3 clear bands within that gradient. Okay, so that's what density gradient centrifugation is. But this is an experiment about DNA. How are we going to use this to separate out different types of DNA? Okay, so this is where the special way they grew E. coli comes in. Okay, now you do need to remember some physics for this, right? If you came into A-level biology thinking you were going to be able to forget all your chemistry and your physics, well, that is completely untrue. Physics is a wonderful subject, incredibly useful, and you should remember every scrap of it that you've been taught because you never know when it will come in useful. So you should know what an isotope is. Basically, it's a version of an atom with a different mass, a different atomic mass. So usually same protons and electrons. So in terms of its chemistry, the atom behaves in the same way, but an extra neutron. So in this instance, we've got normal nitrogen here, what I would call normal nitrogen, it's nitrogen 14, right, which has got an atomic mass of 14. As you can see here, look, the key there, proton, neutron, electron, but it's the nucleus we're interested in here, look. So in terms of protons, 1, 2, 3, 4, 5, 6, 7 protons, and then 7 neutrons as well. That is standard nitrogen. We've then also got this version here, nitrogen 15, which has still got those 7 protons, but can you see that little extra neutron down there? Okay, and that's what makes it nitrogen 15 as opposed to the normal nitrogen 14. Okay, it's a slightly heavier version. Okay, now all organisms, all biological organisms need a source of nitrogen in various molecules, one of which is of course DNA. Okay, DNA involves nitrogen. So if you can feed bacteria different versions of these different isotopes of nitrogen and get them to incorporate them into DNA, you'll end up with some DNA that's slightly heavier than normal DNA. Okay, and the way you do that is through an agar plate. Okay, so here we've got standard agar plate with standard just blobs of E. coli going on here. Now agar, as you should know, is just a jelly, right? It's a gelling agent, and you can mix into it whatever you want, right? Normally it's standard nutrient medium that gives the bacteria everything it needs to grow, a source of carbohydrate, a source of nitrogen to make proteins and DNA and so on and so forth, and all the vitamins and minerals and water that they need. Okay. Now what Meselson-Stahl did was they took a nitrogen-containing molecule, in this case ammonia, right, and mixed that into the agar, pretty standard, okay? And what they surmised would be happening would be the nitrogen mixed into the agar jelly would make its way into the nitrogenous base in the DNA, right? Pretty logical. So the bacteria would use the materials they were providing in the agar jelly to make its nitrogenous base. Okay, same thing. But this time, if you use the heavier version of nitrogen in like a heavy ammonia, right? So they mix the ammonia with nitrogen 15 in there, then of course it would be that heavier nitrogen that would make its way into the nitrogenous base. So the DNA would be slightly denser, slightly heavier. Okay, so that's basically what we're talking about. So first off, they then did this on 2 separate plates, one with nitrogen 14, one with the nitrogen 15, and essentially sort of grew them and then put them in their density gradient centrifugation to see if they could tell the difference. So what we've got here is bacteria that's been grown for a long time, lots of generations, on nitrogen 14 ammonia containing agar. So they then take the bacteria off, bust them up, extract the DNA, very easy practical, some of you may have done it, take the DNA out, stick it in the density gradient centrifugation. Now of course they didn't use sugar, they used cesium chloride here, so lots of cesium chloride down the bottom and less at the top, and basically spin. Now nitrogen 14 ammonia fed bacteria produce DNA that stays pretty near the top. We'll call that light DNA. Okay, it doesn't push very far into that density gradient at all. But when you take bacteria off the other plate, the one with the nitrogen 15, okay, well then what we get is heavy DNA, which pushes much further. Okay, now what Meselson-Stahl realised from this is they could actually control what ended up in the DNA, control the density of the DNA via what they fed the bacteria on the plates. Okay, so what they then did next was go, okay, if we're going to grow bacteria for a long period of time on here, right, on this heavy DNA, we know one generation takes 20 minutes. So the bacteria will have gone through one step of replication on average in 20 minutes. Okay, so we take our bacteria out of here, okay, out of this heavy nitrogen containing medium, okay, and place them in another medium. Now this could have been an agar plate or a broth, it doesn't matter. I'm sticking with agar plates because you're probably going to be more familiar with them. Okay, so take that off there, pop them in the light nitrogen containing medium for 20 minutes, kill them, get the DNA and see where it ends up. Okay, and what they saw was that. Okay, so all the original DNA is heavy. They then give the bacteria the opportunity to replicate once, and they don't get heavy DNA or light DNA, they get something in the middle. Now those of you that have paid attention to the semi-conservative replication lesson will instantly see why that's the case. Okay, because one strand, right, was from the parent, made on that heavy nitrogen, right, and then one strand in that one replication was made on the light nitrogen. Okay, so each double helix is made of one heavy parental strand and one light new strand. Therefore, the overall double helix held together by those hydrogen bonds is somewhere in between. Right, is an intermediate DNA molecule. Okay, so that immediately eliminates conservative replication as a possible solution, okay? Because if conservative replication were true, what was going on, what would you expect to see is you would expect to see the original double helixes down here, all heavy, and the new ones up the top there. But you don't see that. You see an intermediate DNA, okay? So at this point, everyone's like, okay, fair enough, it's semi-conservative, okay? We're now very confident that semi-conservative, as we thought, is what happens, most likely in the first place, and it is what happens. However, we can't completely discard dispersive replication, because dispersive replication would have given that same result. If you cut, right, the DNA strand and just put in a few chunks, or 50% of the chunks from new light DNA, light nitrogen, then you would see the same result. So we need a second step, okay, to disprove dispersive replication. And in this step, all they did was grow the bacteria on the light nitrogen for 2 generations. So after 1 generation, we got that, okay, so we've got each strand has got an old and a new. Then we're going to let them grow for a second. So those split up and are used as templates. Now, a lot of you, I hope, will be able to immediately see what is going to happen in this tube, okay, in the second generation. Okay, so if not, pause the video and have a think about it, because you should be able to work it out. It's a pretty standard question. Okay, if you've got a parent double helix which has got one heavy, one light, and they both split apart and they're both used, all the new stuff is light, what band or bands are you going to see in the second generation? Okay, and the answer, of course, is this, right? You see an intermediate DNA molecule, okay, because you've still got that original heavy DNA there, okay, it's still there, right? It has been used as a template, okay, and a new light strand's been built next to it, but this time that light strand you made in the first generation is also being used as a template. Okay, so what you end up with is now 2 that are light. Okay, so what we've ended up with there, it cannot be dispersive replication, because if we were just adding more light DNA in and diluting every time, what you would see is you'd see half the first time, 1 band at 3/4 of the way up, and then an 1/8, and then a 16th, and a 32nd, and so on and so forth. And you'd be diluting out the heavy DNA. You don't see that. However long you do this for, you still see that intermediate DNA band, because that heavy DNA is still being used as a template in each generation. Okay? But what happens subsequently is that the new light strands are also used as templates, and you get these double light helices as a result. Okay? So that eliminates dispersive replication and leaves only semi-conservative replication as your last and certain point. Okay? Now that is a standard question to ask on an AS level. Look at these tubes, explain the results, what's going on, how do we know it's semi-conservative replication, what does this show, what does this prove, and it brings in loads of other practicals, okay, from across the AS level, okay. So it's a really crucial thing that you get your head around and understand, okay. All right, I hope that's clear. That's DNA replication and how we know done. Next, onto a modified form of nucleic acid, ATP. I'll see you then.